Functions of proteins. There are thousands of proteins or millions of proteins which are existing in nature, which are present in several cells, eukaryotic cells, prokaryotic cells, as well as in viruses. So these proteins are responsible for carrying out several functions, and these proteins have different roles in different types of cells or viruses. So let us try and understand different examples of proteins and their functions in a biological cell or in viruses. So these functional category of proteins, the functions of proteins are divided for our convenience into different categories. So let us see all these functional categories of proteins. So the first functional category of proteins is catalysis. Catalysis, yes, proteins are involved in catalytic reactions. And we have already learned about enzymes, that enzymes are the proteins having catalytic activity. And these enzymes, as their natural role is, they enhance the rate of chemical reaction. So whatever the reactions, biochemical reactions that happen in a biological cell, all these biochemical reactions are catalyzed by enzymes, and enzymes are proteins. There is one exception, that is ribosome, which is made up with nucleotide. All the other enzymes are made up with proteins. So enzymes are involved in carrying out biochemical reactions, that means metabolic reactions. They are anabolic reactions or catabolic reactions. So that is the first function of protein, that they are involved in catalysis. Second, proteins serve as a storage of energy and carbon source. So proteins can be stored in the cell as a reserve food material or as a source of reserve nutrients. That means whenever there is depletion of nutrients or there is unavailability of nutrients in the surrounding of the cell, then this reserve stock of proteins can be used by that cell as a source of carbon and energy. That is the meaning of reserve food or nutrient. So X proteins in seeds of examples are the proteins which are present in the seeds of wheat, corn, rice, albumin protein which is present in egg white, casein in milk. All these are the examples of storage proteins, and all these wheat, rice, albumin, egg white, all casein, all these are the sources of this bulk protein source. Then the third function of proteins is proteins are involved in giving structural integrity to several cellular components. We call them as structural proteins. So these structural proteins are responsible for giving strength and protection to different cellular components and hence to the cell. Examples: collagen. So collagen is a protein which is present in the skin, hairs and feathers. Leather is a pure collagen. So we have already learned about this collagen protein, that it is a strong fibrous protein which is an essential component of skin, that makes leather, which is complex with other polymers also. So collagen is an example of fibrous protein which is an essential component of leather or skin. It gives structural strength to feathers as well as hairs. So these proteins are strongly hydrophobic proteins which makes the structures strengthful and hence responsible for protecting the cells. Next example of structural protein is flagellin. So flagellin is a protein which is present in bacterial flagella. So flagellum is a component of bacterial cell which helps in the motility of the cell. So this bacterial flagellum, it consists of, if you see the structure of this bacterial flagellum, it consists of filament, then a basal body and hook. These are the three components of bacterial flagellum. So this filament filament is made up with flagellin protein. The filament of bacterial flagellum, it is made up with flagellin protein. So these proteinic subunits are arranged in an order such that they form a hollow cylindrical or helical shape of filament. This is the structure. This is the cell wall of bacterium, and this is the plasma membrane at the bottom. Then this is the flagellum. So this is the basal body, which consists of rings. Then there is a hook, and then this is filament. So you can see that this filament is made up with proteinic subunits. So these proteinic subunits, that is flagellin protein. So these flagellin proteins are bound to each other, connected to each other in a regular fashion in order to form a complete filament. So that is the example of structural protein in bacterial flagellum, that is flagellin. Another function of proteins is they are involved in defensive mechanisms, particularly in mammals. So role of proteins in defensive mechanisms is because of the presence of immunoglobulins. So immunoglobulins are the glycoprotein. So it is a conjugated protein. Glycoprotein means it is a protein which is conjugated with carbohydrate. That's why that is called as glycoprotein. So an example of glycoprotein which is involved in defensive mechanisms in the mammalian's body are immunoglobulins. So as the name indicates, immuno, that means these are the proteins which are involved in defensive mechanisms. They are involved in immunity or immune response, and that is why they are called as immunoglobulin. So they are involved in defensive mechanisms. So they are glycoproteinic in nature, and it consists of a globulin protein. They are generally called as antibodies. So we know that antibodies are responsible for the neutralization and elimination of foreign bodies. Foreign bodies such as bacteria, viruses, fungal spores, protozoa, etc. etc. We call them as antigens. Any foreign substance which when enters in our body gives a response in the form of production of antibodies, and that substance is called as antigen. So when antigen comes in contact with B lymphocytes, these B lymphocytes proliferate and differentiate into plasma cells, and then these plasma cells secrete antibodies, that is immunoglobulins, and then these immunoglobulins bind to antigens, and binding of antibodies to antigens, it is called as neutralization reaction. So all the sites on antigen are blocked or occupied by antibodies, and that is why this reaction is called as neutralization reaction. So they are responsible for elimination of foreign substances like bacteria, viruses, different parasites, etc. etc. So they are involved in defense. Now let us see the structure of this defense protein, that is immunoglobulin. We need to keep this in mind that all immunoglobulins are not antibodies. All immunoglobulins are not antibodies, but all antibodies are immunoglobulins. This means that all immunoglobulins which are produced in body may not function as antibodies. So that statement is very important, that all antibodies are immunoglobulinic nature, but all immunoglobulins may not act as antibodies. That means they may not be involved in neutralization of antigens. So this antibody molecule, it is Y-shaped, and this Y-shaped molecule, it is flexible at the central region, means where this junction is there at Y, this junction is there, so there is antibody molecule is flexible, and that region where it is flexible, it is called as hinge region. Hinge region. So immunoglobulins are of different types. They are basically divided into 5 categories. Antibodies are of 5 types. IgG. Ig stands for immunoglobulin type G, immunoglobulin type M, immunoglobulin type A, immunoglobulin type D, and immunoglobulin type E. So these are the 5 types: IgG, IgM, IgA, IgD, and IgE. These are the 5 different types of antibodies, and these antibodies are responsible for different functions in different secretions, or they are responsible for carrying out different functions at different locations. For example, the primary antibody which is produced after the entry of antigen are IgM type. So primary immune response leads to production of, first that leads to production of IgM, and later on there is production of IgG type of antibodies. So if the antigen is entering in our body for the first time, there is production of IgM followed by production of IgG. If the same antigen enters in our body for the second time, there will be production of IgG type of antibody. So this IgG type of antibody is the principal antibody which is produced in our body against different antigens. Then IgA, this antibody is generally present in our secretions like nasal secretions, saliva, etc. IgD, these antibodies are produced during allergic reactions, and also IgE are produced during allergic reactions. Each class of antibody has different role, as I mentioned. So in general, these antibodies protect our body against circulating bacteria, viruses. They are responsible for neutralizing the toxins. They are responsible for activating the complement system. Complement system is again responsible for activation of a cascade of lytic proteins that brings about the lysis of a bacterial cell. And when these antibodies are bound to antigens, they enhance the effectiveness of phagocytic cells. That means if a bacterial cell is bound by antibodies, then this antigen antibody complex or bacterium antibody complex, it can be engulfed very easily by phagocytic cells such as macrophages. This is the structure of antibody molecule. It is Y-shaped, and it consists of 4 polypeptide chains. Out of these 4 polypeptide chains, 2 are identical light chains, as it can be seen in the figure. These are the 2 light chains. Then 2 heavy polypeptide chains. So the names are given depending on its molecular weight. Light chain has low molecular weight, heavy chain has high molecular weight, and that is the reason why they are called as light chain and heavy chains. So 2 identical light chains and 2 identical heavy chains. So that makes this Y shape, and all these chains are bound to each other through this linkage, which is called as disulfide bridge or disulfide linkage. This linkage is the same as we have learned in the formation of tertiary structure of protein. So here there is presence of sulfur containing amino acid like cysteine. Here there is presence of sulfur containing amino acid cysteine. So it has CH2SH group, CH2SH group. So there is removal of one molecule of H2, and it leads to formation of SH, that is called as disulfide bridge. So all these chains are bound to each other through this disulfide bridge. This region is called as hinge region, which is flexible. Then this is the site where antigen binds. So it is called as antigen binding site. So this site and this site are identical. So this is the hinge region, which is flexible like this. So according to the structure of antigen, it can adjust its position for antigen binding site for the binding purpose with antigen. Next example or the next function of protein is their involvement in the blood clotting mechanism in mammals. So proteins such as fibrinogen protein, thrombin protein, these are the blood proteins which are involved in formation of clots. So whenever there is any injury, in order to prevent the loss of blood, in order to prevent bleeding, there is formation of a plug. That site is plugged or it is blocked in order to avoid the bleeding. We call it as clot. So these are the proteins which are involved in formation of clots that prevents blood loss from body. Then the next function of protein, they are involved in regulation. So there are proteins in a mammal's body or in plants which are responsible for the regulation of metabolism. So regulation of metabolism, these proteins are called as hormones. So hormones are the proteins which regulate physiological and cellular activities of the body. A very common example is insulin protein. So insulin is a protein which is present in mammals body that regulates blood sugar level. So insulin is a hormone which is produced by pancreatic cells, and this insulin, it helps in the transport of glucose inside the cell, absorption of glucose inside the by the cells. If it is not there, then its deficiency leads to diabetes, because the blood sugar level increases because it is not absorbed or it is not being transported to the cells. Then proteins are involved in preventing the freezing, that is they show anti-freezing effect. So an excellent example is an antiphase protein present in Antarctica fishes, which are present in Antarctica region where the temperature is below 0 degree Celsius. So these proteins are responsible for protecting the blood of these animals from getting freezed. So even after exposure to temperature below 0 degree Celsius, the blood never clots, and the mechanism behind that is the presence of antifreeze proteins. Proteins are also involved in transportation. They are called as transport proteins. We can see the structure of bacterial cell cytoplasm. So that cell cytoplasm, it is basically made up with phospholipid bilayer, and that phospholipid bilayer we call it as a fluid mosaic model, as we have learned earlier. So this fluid mosaic model, that is the phospholipid bilayer, it contains several proteins in between. So they are responsible for the selective transport, or they are responsible for formation of channels. So you can go back and see the structure of phospholipid and the structure of cell membrane. Another example is hemoglobin. So hemoglobin is a globular metalloprotein. Why it is called as a metalloprotein? Because this is a protein which is a type of conjugate protein which is conjugated with metal. So metal plus protein called as metalloprotein. So the function of this hemoglobin is to carry oxygen and CO2, that is to transport molecular oxygen from lungs into tissues and CO2 from the tissues back to the lungs. So this transportation is the function of hemoglobin, transport of oxygen and CO2. The structure of hemoglobin, we have already learned that it has a quaternary structure. It consists of 4 polypeptide chains, 2 identical alpha chains and 2 identical beta chains, and all these chains are bound to a heme group, and this heme group is the one which contains Fe or iron, which is responsible for binding to oxygen and CO2.